Welcome to the Jack Westin MCAT Podcast with your host, Phil Hawkins.
And Azaii Calderón Muñiz.
Alrighty, excited for yet another very structured, very fun podcast. So today, you may not be able to guess it just from that little hint, but we're going to be talking about the cytoskeleton, the structure, the backbone of your cells. So we want to think of, we want to understand, yes, what is the cytoskeleton? What are the components of the cytoskeleton? Also just why it's important to know about it. And of course, beyond why the MCAT, right? That the fact that the MCAT's going to test you on it. Yes, that's important enough to make it worth learning about, but also just what does it do, right? So before we even dive in, taking a step back, the cytoskeleton. So like I mentioned, the structure, the backbone of your cells, because obviously you don't have an entire actual skeleton inside each one of your cells. If you do, we need to have a conversation. Mm-hmm. Now, the cytoskeleton is essentially just this series of filaments or fibers that are inside your cells, primarily actin, which are also known as microfilaments, microtubules, which are known as microtubules, and intermediate filaments, they don't get a special name. And so their primary differences are going to be things like size, but also what they do, their actual structure. So the cytoskeleton is involved in a whole lot of things. You're going to hear us talk about what it's involved or what the different filaments are involved in. That can be specific to each, but recognize that there will be some overlap between them.
Yeah, there's a lot of differences, and we're going to talk about each one of these kind of individually, as well as even a couple of things that people think are cytoskeleton, but they're not cytoskeleton. Just kind of like clarifying some of that. But we're going to talk about actin first. It's the smallest filament. It's also all over in your body, right? It's somewhere around like 10% of the proteins in your body, plus or minus some amount. And so it's huge, right? Like the actual number of proteins in your body, like 10% of them are just actin. Now, actin can exist in two forms. And actually, taking a step back, proteins in general often exist in like a couple of forms, either a filamentous form, where it's like, kind of a long filament or string or rod, or a globular form, which means like kind of like a glob, just like a little like wad of protein. And this is actually something that you've probably heard of, but you just didn't internalize. Like if you're like, oh, I didn't know globular proteins were things like that. But like hemoglobin, that's a globular thing. That's why globin's in the name of it. Myoglobin is globular. And this is like proteins with glob in them just mean that, oh, they're just like a little wad of protein that kind of floats around.
Don't you love when like things actually tell you what they are just from their name? So nice.
Yeah, it is. It's a little tricky because I feel like the further I went in medicine, the more and more often that happened. But it definitely would benefit to have some knowledge of Greek and Latin to be able to get things. Because sometimes it's like, it literally is translating a foreign language. So globular just means a little like wad. And so actin can exist in a globular form or an filamentous form. Very often we just call it G-actin or F-actin for globular filamentous. But like when we have like our little actin monomers that are just floating around inside the cytoplasm, that's globular actin. When it starts to link up with other ones and create like a filament, that's filamentous actin. There's a whole lot of study on the way that filamentous actin forms and like building it and breaking it down. And it turns out that like regulating that inside your cells has a whole like slew of effects. And like you can actually like tell what cells are going through mitosis, whether an artery is contracting, like vasoconstricting or vasodilating, by just kind of like measuring the F and G actin inside the cell. And so the MCAT likes to ask questions. I know that they've had a passage. I know they have a passage on that on one of the practice exams that are out there. And so this is something that the MCAT it's really useful to just kind of know what is G-actin, what is F-actin, what does that actually mean? Now, kind of like going a little bit, like kind of be honest, if like actin isn't really useful in a globular form, it's got to be in a filamentous form for our body to actually do anything with it. But I'm willing to bet that when you hear actin, if you like think about something in the body, It's probably muscles, right? Muscle contraction. And this is why actin is such a huge percentage of the protein in our bodies, because all of our muscles are pretty much just myosin and actin. Like that's the two like functional units working together that makes muscles contract. So you have myosin that will pull on the actin, which is just like a little rope, if that was a way to think about it. Myosin is not a cytoskeletal thing. We'll talk, it's a motor protein. We'll talk more about those in a little bit though.
Yeah, I completely agree with you. Like everyone thinks, okay, muscle for actin, but there's so many other parts of the body that actin is used in, right? And within the cell. One of the things that you will probably see on test day, if you see actin, could very well be the contractile ring. So when cells are dividing, and I think we actually might have touched on this before in a prior podcast, but when cells are undergoing, mitosis, right? And they are pinching off, it's actin that forms that contractile ring and essentially pinches the two daughter cells apart, right? It's also involved in endo and exocytosis. So opening the cell up essentially, right? Obviously not literally opening it, but so to speak, metaphorically, opening the cell up, bringing things in and kicking things out. And if you think about why that works, right? It's literally, like you were saying, these filaments, these strings. And so if you shorten and lengthen them at different places, what can you do? You can move. So it's also involved in cell motility, because now I know, oh, I'm going to lengthen the ones at the front end of the cell and shorten the ones at the back end, and then keep doing that. And that's going to help move the cell. So actin is involved in a lot of different things that you should be aware of for test day.
Yeah, that amoeboid movement is what they call it, where like basically your actin strands can grow and push out the walls of the cell, or they can shrink and the walls can get kind of like sucked in. And this is how like white blood cells can go, and like macrophages can go and like engulf things and kind of like ooze around between cracks in between cells. And so actin is also useful for cellular movement, like for how cells move overall. This is also how amoeba move. And so this is also like some pathogens utilize actin as a way of locomoting by just kind of like having their cell like membrane bulge out in certain areas, and that's how they kind of like crawl. I always think when I think about the contractile ring. Oh, sorry, Azaii, I could tell you.
No, go for it.
Okay. When I think about the contractile ring, I always just think like having a giant water balloon and then just putting a belt around the middle of it and just cinching it tighter. And so that's how like the cell will split into two. I wish that's how it worked. Like if you had a giant water balloon, you put a belt around it and you pinched it off. Now you have two smaller water balloons. But that's essentially how that like cytokinesis occurs with the contractile ring.
That would be fantastic until it pops. You know, you're just waiting for it, but that's perfect for a water balloon fight. You just like tighten it up really, really well. And then the moment that you toss it at somebody, boosh, whole
just
thing
the pressure.
Yeah, that would be really fun. Okay. Now you've given me ideas that you don't need to have given me. Something that can be really helpful if you're having trouble picturing what we're talking about, right? The contractile ring and pinching off, cytokinesis, being able, cell motility, lengthening, the actin filaments at one end, shortening on the other. Look up, just kind of Google search wherever if you still use Bing or Yahoo, look up actin, microtubule, cell, and then fluorescent, some combination of that. And what you'll see is that people very frequently tag the different cytoskeletal elements with fluorescent tags, and you can see they'll, label whatever color is which, and you can see where they're distributed in the cell. And that can help you understand what their role is if you know where they're hanging out. So that can also be really helpful. It's also just really pretty.
I was just going to say that. There's
so
also
pretty.
Just like really beautiful images.
Yes.
I really want to have a bunch of just like science art on the walls of my house. And like when I see like those fluorescent microscopy things with actin and microtubules, I'm like, oh, like I just want to hang that on the wall, because that's something that should be in a museum.
So our histology and pathology professors in first year, they would just find really cute or like artsy, things that they saw under the microscope, and they would save them. And so every so often during a lecture, they would just include that. Science is beautiful. It can produce some really amazing photographs, images, but I do encourage y'all to look it up. It also just does help orient you as to what you're looking at. Have a more structured understanding of it. All right, organized understanding. I'm done. I apologize. I'll let you keep talking, Phil.
No. I think so. Actin is in like a lot of places. I also want to point out every single cell in your body has actin. Like a cell cannot survive without actin. So it's not just in muscle like stuff. It's everywhere. And so that's a big thing. The contractile ring, the endoexocytosis, the mobility stuff, and then the muscle stuff. Those are the 4 things that I think the MCAT is most likely to test you on when it comes to actin. Note that because actin is in every single cell, often, like if you're trying to measure the rate of a certain protein being expressed, you might also measure the rate of actin as a control, right? To just say like, okay, like the protein I'm measuring, it went down, but regular protein didn't change. Like actin didn't change. And so sometimes you'll see actin as a control because every cell has to have actin in it. And so it's a good protein to measure as a control.
Yeah. Just since you bring that up, when you measure protein or levels of RNA of a particular gene or something like that, you'll often hear it called as housekeeping as a control. So it might be called like a housekeeping gene because it's constantly being expressed. And so just something to have in mind for those of you that might do research, probably makes a little bit more sense. But if they throw something like that out there, recognize why they're doing it.
Yeah, I think the most common things that happens with is actin is one, probably also some of the proteins for metabolism.
Yeah, I think GADPH.
Yeah.
My words.
Yeah, that's it. Glyceraldehyde dephosphorase. I can't remember. I don't think that's right, but GADPH, whatever it is. There is also the other one that they're likely to test is tubulin. Now, I've seen this. We're going to talk about tubulin more in just one second, but I have seen multiple things from the AAMC where like they're talking about some weird protein, and then all of a sudden in figure, they have tubulin or they have actin. And most of the time students are like, wait, what? Like they never mentioned this protein anywhere in the passage, and all of a sudden it's in this figure. Like, I don't understand what's going on. And that's why it's important to understand what's going on with like having those controls, and actin and tubulin are really commonly used for that.
Yeah, absolutely. I think all of this is really important to keep in mind, and it just shows that understanding different components within the cell, right, and what's going on in your body can also have applications outside of just the actual content and help you extrapolate information, right?
Yeah.
So super big, something you will do all the time as you continue to progress in your career. So get used to it and learn to enjoy it.
Yeah. So tubulin, right, is actually the monomer for microtubules, which are obviously another cytoskeletal thing, which we definitely we have to hit. We have to talk about this. Actin, otherwise known as microfilaments, as I said, those are the smallest cytoskeletal structures. Microtubules are the largest, and they're actually tubes, right? Like they're hollow inside. And so that's why they're called microtubules and why it's called tubulin, right? And so think about like that, like a pipe has got to be bigger than a string, right? Mm-hmm. And so they're both made out of proteins, but because one has to create like a tube, that's going to be a much larger structure than the other ones. Now, the tubulin itself, which is the protein that the monomers, it exists in these alpha beta heterodimers. So there's these two proteins that are different in structure. We call it the alpha tubulin and beta tubulin, and they come together. And then that functional unit just gets repeated over and over and over to create the entirety of the microtubule. Now, microtubules function, what are they used for? The first things that should probably come to your head is just movement of chromosomes, and your the what do they call it? The spindles, right? I need more coffee in the morning. It's like the spindles, the cell spindles, when you have like mitosis and meiosis, those are like sometimes called the microtubule organizing complex, or MTOC. And so those spindles, how those work is they got microtubules just growing out of them in all these different directions. And so it allows you to like grab certain things and pull, because you need to have this reference in order to pull and push things in different directions. And so that's where the microtubule organizing complex comes in, which is useful in all the like the cell division stuff.
Yeah. And it's actually really cool because the same way that it can help promote the movement of chromosomes within the cell, it can also promote the movement of other intracellular important components. And so there are going to be motor proteins that are involved here and that interact with it. I really quickly want to make sure, because I don't think we already mentioned this, there are associations with ATP and GTP for both actin and microtubules. And so with actin, it's helpful ATP, right? So there's ATP binding. With microtubules, it's GTP. And so you might hear them talk about this GTP cap. And it's essentially a way for the microtubule to avoid kind of coming apart, right? And you need this ability to quickly promote elongation and then shrink, right? Because if you're going to be moving entire chromosomes, you have to be able to go back and forth. But how do you make sure that you don't just make it go so long that it ends up at the other end of the cell, right? Or that it just completely degrades and now you have a one heterodimer stump of a microtubule. And so
I just want to pause that like breakdown. That's one of my favorite terms in catastrophe. My favorite terms in cell biology is like when the microtubule starts to like break down, like the actual technical term for that is catastrophe, which is like, that's such a, like, I feel like whatever scientist was like named that, like he was having either a really bad or a really good day. And it just like pushed him over the edge. It's like, oh, it's catastrophic. Like my research is over. I'm calling this catastrophe.
Well, that may be supported by the fact that when the GTP cap is recuperated, it's called rescue. So, so it's a catastrophe and they're rescuing their work. But yeah, so it's really helpful and important to, have this in the back of your mind, because it's just another way that they can test you, another set of questions that they could ask if they have a passage with microtubules or actin. Right now, nucleic acids are on the table, energy production is on the table. So remember, the MCAT, yes, they're going to care about the content that you've learned, but also the connections you can make. It's all about relationships, which is why sometimes we sound like we're talking about a bajillion things at once, and it's because there sometimes are a bajillion things going on at once. So back to microtubules. So Phil, you already talked about their structure, right? Alpha, beta heterodimers. You talked about chromosome movement and the microtubule organization centers. So we've also now talked about the just kind of intracellular transport, but similar to actin, they can be involved with movement. And so the cilia, the flagella, right? The cilia, in your respiratory system. Trying to find the word. I couldn't find it. There we go. Are also going to require constant movement, right? When you are coughing, and this is going to be a little gross, but when you're producing mucus because you've had COVID and you're coughing, right? That productive cough, something has to bring it up, right? Not just the diaphragm contracting, but you also need like something to physically move things up. And so the cilia are fantastic at that. And microtubules play a big role in this. And so the same way that, actin can be involved in movement, microtubules love movement, and they're associated with intracellular movement of important products. Yeah.
You should just think movement with microtubules. I want to take a second, like talking about what happens in like the respiratory system. I actually think that's a very likely way for the MCAT to try to test you on microtubules specifically. And so like we have this is another just great term that like I'm really excited about. It's kind of gross actually now that I think about it, but like we have this thing called the mucociliary elevator. And maybe that makes you think of just like a weird gross elevator that's like filled with mucus, but that's kind of what it is. And so when you inhale like particles like dust and smoke and things like that, those little particles will get caught in the mucus that lines your respiratory tract. And that's cool because that means they don't end up in your alveoli and they don't mess with your ability to breathe overall. The thing is, like, we can't just keep piling up all that junk in there forever because your lungs would fill up. And so what happens is, like, in your bronchioles and trachea, you have cilia, which do this kind of like wave thing. It's like the walls are covered in hair, essentially, and all the like little hairs just push up over and over. And so the mucus, which is holding all the like dust particles, gets carried up your respiratory tract and then dumped into your esophagus. So
Someone's having a small heart attack right now.
Yeah, I know. It's like, that sounds really gross. Like, wait a minute. There's all these, not only is it filled with mucus, but there's tons of tiny hairs that are whipping around all the time. It's actually kind of cool, but eventually all that stuff ends up in your digestive tract. And so like that way you're not like filling up your lungs constantly. But that mucociliary elevator isn't like a thing kind of going on with that. You also mentioned flagella. So like sperm motility, like what's going on with like how sperm move. Obviously that's going to be a flagellar movement. So I think it's really good and really important for you to have examples in your head of these things, right? Like, what are the cytoskeleton? Because actin is also for movement, right? But actin is for that amoeboid, like oozy movement that we see with like white blood cells kind of like going and expanding, like macrophages, versus like a spermatozoa. Like that's going to be more of a flagellar thing. I also just like with the intracellular thing, I always think about them as like highways, right? Like there's just like highways inside your cell, like going from the nucleus to the micro or to the Golgi, to the endoplasmic reticulum, to the lysosome. And like all those highways are just microtubules. And so we have, as you mentioned, motor proteins that'll just kind of like carry stuff to and from the different areas. I think about it as just like semi trucks driving around inside your cells, just like delivering stuff to different places. That's how I see those microtubular motor proteins.
Yeah, I'm not ready to have an entire semi truck in one of my cells. Got to shrink it first.
Yeah, just a small one, right? So we talked about actin, we talked about microtubules, and the third one that everyone always wants to talk about is intermediate filament. And maybe I was just a really bad student and I somehow didn't figure this out until way too late. I was in med school before I figured this out. So you guys are going to be better than me. But intermediate filaments are not a single type of filament. Intermediate, like we have actin, we have microtubules, and intermediate filaments just means anything else. Like other is what intermediate filaments mean. So there's like 33 types of keratin, which is an intermediate filament. And like there's 33 types of just that one intermediate filament. And there are tons of them. Desmin, vimentin, I have a list here. Peripherer, syncoilin, internexin, neurofilament, cynemin, lamin, like gliofibrillary acetoprotein, right? All of these are intermediate filaments. And so there are tons of these. They're all over the place. And what intermediate filaments are in a certain cell might be different in a different cell. So keratin, for example, is something that we see tons of. Like I always think like keratin is like Kevlar. Like keratin is like very tough structural protein. And that's actually what we have in our skin that makes our skin really hard to rip and tear compared to other parts of our body. Right? Like if you took your thumbnail and you ran it along like the liver, like it would actually like rip and tear the liver. But if you do that to your like skin, it's not going to do it to your skin. And that's because keratin acts like kind of like an armor plating thing. Keratin's also what we make our hair out of. And our fingernails are made of keratin. So obviously they can be like very strong and structural. But so we don't see keratin in all cells, but we see it in a lot of like exterior epithelial cells. Keratin are way more common in that. And so that's actually a really interesting thing because that's actually one of the ways that you can diagnose a certain type of cancer. Like if somebody has cancer that is metastasized, and it's in the liver and the lungs, and like maybe it's in the bones as well. You can go in and take a biopsy. And if I go in and take a liver biopsy of this cancer, of this tumor, and then I like do some like analysis of that tumor, if I find that tumor has a whole bunch of keratin in it, then that's probably a skin cancer that has metastasized and spread throughout the body. And so you can actually use intermediates to help diagnose and figure out what type of cancer something is by figuring out which of these strange filaments tend to be really common. Like desmin and vimentin are in a lot of muscle cells. And so there's a bunch of just like, depending on which ones are present, you can kind of figure out, okay, what kind of tissue was this supposed to be?
Yeah. One quick kind of associated thought is that these can be involved in, like attachment to the matrix and whatnot, and different types of, like, for example, desmosomes, hemidesmosomes. You need to know all of that. So you need to know the difference between a desmosome, hemidesmosome, tight junction, gap junction. So we're not covering that, but we want to make sure that you do go and look that stuff up. And remember, we're trying to remind you what you need to know and what to study and what the relationships might be, because they're all fair game if they're related on the MCAT for a given passage.
Right. And it's really interesting. I mean, what always comes with these sorts of conversations, and this is a good question that you guys should be asking, is like, why do I need to know this as a physician? Right. And I know we've kind of talked about like a lot of things kind of relating to this as well, but there's lots of treatments that focus on these things as well. And so we've already talked about how Actin is and microtubules. So those two specifically are critical for every single type of cancer, or for every single type of cell. Sorry. But specifically, Actin does a lot of stuff with the actual cytokinesis, right? Like forming and like splitting into that, like division into the two daughter cells. And microtubules have to do with moving chromosomes. So if you give somebody a medication or a drug that messes with microtubules, it's going to be harder to actually like go through mitosis and meiosis, because you can't push and pull and move the chromosomes. And I know that there are cancer medications designed to do that, to like disrupt microtubules as a way to slow down cell division and like inhibit the growth of cancers.
Yeah, I had to make sure, because as you were talking, it gave me a throwback to undergrad. And I don't know if some of you guys have also done this in your classes, but they will give you slides and they will show you cells that have been treated with some of those medications. And Taxol came to mind. I was like, I have to make sure that this is what I think it is. And Taxol, by the T, can confirm. It's a microtubule, and this is going to sound weird at first, but hear us out. Promoter and then stabilization agent. If you think about it, cool, it's elongated, but it's stuck. And so it can't do its function. And so if it can't do its function,
the catastrophe, which is important for like pulling things back apart. Yeah, exactly.
And so there are medications that, people are using and that people are taking, and that are being used to study as we continue to learn more about different illnesses and in cases of cancer. Now, some students are like, well, I'm not doing pharmacology, so since I'm not going to be a pharmacologist, maybe I don't actually need this. You still have to understand how these drugs work. I was in a clinic with seeing a couple of patients with cancer, and I was working with some physicians that they see a ton of patients with cancer all of the time, and they know the mechanisms of the drugs that they are giving. This isn't just some like, oh, leave it to someone else to know. They know the mechanisms that helps them understand the side effects that patients are going to experience. And when they see the patient each time, they're asking about those side effects. And the same goes for PCPs, right? If you know your patient's on a certain medication, you need to know what the side effects are, and knowing mechanisms of action are key. Let me also tell you, your preceptors, the doctors you'll be working with, will ask you questions about side effects all the time. They will also ask you, and this is something that I think is really easy to kind of forget. They'll also ask you, what receptors is this medication, are these medications working on? Things like that. So make sure that you come into this with an open mind, because there's something that you can learn from just about every section on the MCAT, not just content, but about how you learn. So I think that's really important and really easy to forget as well.
I just kind of want to point out, because this could be like sort of a question on the MCAT actually, but like if somebody was on a medication that inhibited or messed with microtubules, right? So there's going to be lots of different effects of that. But one of them, like if you're thinking about like sperm production, right? And like how sperm work, right? Like obviously messing with the microtubules is going to mess with the division itself. And so the sperm count's going to drop for sure. But also because the microtubules are messed with, the sperm motility is going to be messed with a ton as well. And so if you have somebody who has like skin cancer and you're treating them with something that messes with microtubules, like all of a sudden, if it's a male, like they're not like going to be able to have children during that time because like what's kind of being messed with in that case. And so it's just kind of important to be like thinking about like, how are these things all related? Because especially with cancer, a lot of times I feel like that's maybe I'm overblowing this in my head, but I feel like I have ran into several cancer patients that once they are going into cancer, all of a sudden they're like, oh my God, I want to have children and like things like that. And so like putting those, like understanding how these things are relating to each other that's a conversation that I've seen several times, just kind of like going through like, how is this related to these other things? And so understanding those side effects, understanding how this thing works is big.
Yeah. There's so much that goes on that as pre-meds and even as early med students, we don't fully understand. And it's with time that we come to appreciate them. And so if ever y'all are listening, you're just like, all right, here they go again, right? We just don't want you to come in with the same blind spots that we came into this with. And the more we know, the better. One thing that we are all learning, in case we don't already know, is that there are a lot of things that people think are part of the cytoskeleton that actually aren't. And so there is one in particular, collagen, that is often people are like, oh yeah, so cytoskeleton and collagen. Collagen's not inside the cell though. You've probably see collagen in a bajillion supplements. You know, we were talking about my matcha habit earlier. I saw matcha with collagen and I was like, ooh, I have to try this. It's not good. It was not good.
It sounds gross.
Yeah. But, you know, there are a lot of people that are like, oh, collagen, like it's the secret to staying young and whatnot. It's fantastic. Doesn't taste great to drink it, but so be it. But collagen is actually not part of the cytoskeleton. It's extracellular, but it's also part of the reason why people, you know, they're so, oh my gosh, it's fantastic, is that it makes up a huge part of your body. So about a third of the proteins in your body as a whole are collagen, right? So that's huge. And again, you know, you think about, okay, they're filling supplements with collagen, right? They're telling you having this is going to help you stay young or build muscle or whatever. And it's off of this basis that it's protein based. So you've heard us talk a lot about different, you know, what is the cytoskeleton, what is not, right? The filaments that constitute the cytoskeleton, the ones that do not. We kind of left off, though, a very important thing that we said we were going to talk about. And I'm really excited to get to talk about this because it's what actually uses
right. And this is that motor protein portion. And so there are 3 motor proteins that you have to know for the MCAT. I just want to be like really clear on this. Like, this is not like it's just interesting. I mean, it is, but it's also insanely like high yield. So the 3 motor proteins you need to know are myosin, myosin, dynein, and kinesin. So myosin is a motor protein that works with actin. And that's why we were saying, like, you know, muscle growth or muscle activity, right? Like that's actin and myosin. And so myosin goes with actin goes with myosin. Myosin just pulls on actin, and that's how we get muscle contractions overall. Dynein and kinesin, on the other hand, they work with microtubules. And so they will just kind of like walk along a microtubule. Those are the semis that I was talking about earlier, like inside your cell. Like, let's say you just had a macrophage endocytose a bacteria, and now you need to get that bacteria to the lysosome so that you can like kill the bacteria in the lysosome, right? How is that moved inside your cell? It's moved via dynein and kinesin, just walking along these little microtubule highways. The like I note that I keep saying walking along and like doing this little like, movement here. And that's because they actually do walk. They have two legs. So they have like one leg that'll reach and grab, and then the other leg will reach and grab. And then they'll just, they're like actually like walking along. It's like climbing a ladder, how they move like around this. And so it's actually kind of funny to to watch. If you ever can see like a video of like the functioning of those units, it's like, oh my gosh, they're literally just walking along this.
Have you heard of those like Nordic walking poles? They're essentially these like sticks that people use to walk, and they're pretty tall. So
I feel like I use them a lot for hiking. Are we talking about the same thing? I'm just thinking like those like hiking things, or like ski poles. Just kind of like using those.
I think we are. My patients, the ones that come to mind, they're just older, and so they're using them to walk in general. But so the dinings kind of remind me of those poles, because they're just kind of long and dangly.
Yeah.
But y'all should definitely look up the dinings and the kinesins. They like actually walk. They step. Yeah. Like Phil was saying.
I think a lot of times students are like, wait, why is there two? Right? Like, why can't you just use one? And that's because, so a microtubule has a direction, right? So first off, just kind of like taking a step back, because this can cause some confusion. So microtubules are like these, like made of these like alpha beta tubulin dimers that they just stack over and over and over. But even though they're made of entirely the same thing, doesn't mean that both ends of the microtubules look the same. I always like to use the analogy of like Legos, right? If you have like 100 Legos and you stack them up, one side is always going to have the bumps on it, and one side's always going to have like, the bottom with the holes on it. And it doesn't matter how many you stack, the two ends are going to be different. And that's kind of what it's like with microtubules. So microtubules have like one end that we call the plus end, and one end we call the minus end. And so it turns out that dynein walks towards the minus end, and kinesin walks towards the positive end. And so they walk in different directions on these things. And so if you need to move it to one end, you need dynein. If you need to move it to the other, you need kinesin. And so that way you can have things kind of like move in both directions on this highway.
Yeah. And the dyneins will often move cargo like towards the center of the cell. So the way I remembered kind of which direction they moved in is dyneins are done for the day. So dyneins, they're going from the plus end to the minus end, and they're going towards the center of the cell because they're done for the day. Note that these are both using ATP. And so again, this is another place where they could ask you about ATP, energy production, nucleic acids, all those beautiful things. So when you're going through, constantly look for those connections. But hopefully we have convinced you of the importance and the beauty and the fantastic nature of the cytoskeleton, because I'm more convinced than I was before we started this.
Yeah. There's one last thing that we kind of wanted to talk about today, and it's actually not a cytoskeleton thing. Those of you who know Jack Westin and know us, because we talk about this a lot, you know that like we care a lot about lowering barriers to making it easier. We'll never make it easy, but we'll make it easier to go through this pathway and kind of like make the connections and, help you be a better physician. And that's something that we care a whole lot about. And that's honestly, like Azaii and I used to work at like other test prep companies. And the reason we're here is because of that, is because we want to help students first, which is why we do all the free stuff that we do with like, free questions every single day. We do our free trial sessions. We do actually like free lectures, like a bunch of them every week. We just did 6 of them this week, and we're doing 7 lectures next week for free to the public.
Free meetings with our academic advisors.
Right.
Our fantastic resources, y'all.
Yeah, you can meet with somebody for an hour for free, and they will just walk you through like what things you should be thinking about, how to come up with a better game plan. They can even help with stuff even after the MCAT with trying to figure out what resources you need to like help you with interviews or help you with your applications, your primary statements, your secondaries, or even scholarships. I know we had a couple students last year that we helped them with their scholarships, and they got full ride scholarships to medical school. Like their med school's paid for.
Yeah,
right?
that's huge. Med school's not cheap.
That is really huge. And this is stuff that we really, really care about. Obviously, we do have like courses and things like that do cost money, because that's how we're able to pay for all the other stuff that we want to do for you guys. But we know that some of you guys might even struggle for that, to pay for that. And so we're creating a new program called the Ambassador Program, because the other problem that Jack Westin has is people find us when they're looking for practice questions and they don't know about all the other free resources. So we're able to kind of like address both of these things. So if you want to be an ambassador and help us reach out to your college and reach out to your community, to help just like make them more aware of some of the free resources we have, we will, by like going through this, you can get access to a ton of free resources that are normally paid for. Like you can get courses, you can even get 10 hours of tutoring, like free one-on-one tutoring with like an MCAT master, people like Azaii and me, who just eat, sleep, breathe MCAT stuff. And so if you're at all interested in this, right, and getting some of those free resources and helping kind of spread the word, you can email us at support@jackweston.com, but in the subject line, put ambassador, because that'll help us like know what this email is about, because we get a lot of emails. But we would really appreciate that. And I'm really excited to kind of like help get more free resources to students overall.
Yeah. And I realized I don't think we've set this in really a very long time. But if you find our podcasts helpful, interact with them. So like them, rate them, things like that. So that way other students can also see them. And again, it's just one of the many free resources that we provide here at Jack Westin, because our goal is helping you guys succeed. And so just again, reiterating, if you're interested in the Ambassador Program, helping other students and your community get connected with us, and also you getting connected with free resources, then go ahead and send that email to support@Jack Westin, which will be in the description of this podcast. And then just make sure that you have Ambassador somewhere in the subject heading so that they know exactly what you're emailing about. Thank y'all for listening.